medicine, biotech. I had a fascinating meeting with Genentech about 6 years ago, so it's going to be fascinating to see where the technology is advanced, because usually there's some big advancements in the scope of 5 or 6 years. I'm going to keep this introduction short and just welcome you and give you one probably useful bit of information if you have laptops with you. There's an open SSID that's just not broadcast. If you want Wi-Fi, you simply log into the NetApp, lowercase NETAPP network, and you have open Wi-Fi if you need that for your phones, tablets, or almost like your laptops. So with that, I'll keep it brief. I know there's a couple more introductions ahead before we actually get to the main speaker here, so welcome again. I hope you enjoy the content as much as I'm looking forward to it. Thanks.
Thank you, Val. Thank you, Talia. Thank you, NetApp. Thank you, David, for hosting us tonight. Again, it's a great pleasure to be here. My name is Pashu Christensen. I manage partnerships and marketing at The Hive, and I've been also managing The Hive think tank since almost 2 years now. The main information I want to communicate to you tonight is to be online, continue the conversation with us on Twitter, on Facebook, on LinkedIn. Make sure to be part of the community. We have over 5,000, I think it's reaching 6,000 members now on Meetup Group. So thank you for being part of the Hive think tank. We hosted over 60 events, we had over 150 speakers, thought leaders from the Bay Area and the US. We are one of the largest data-driven community in the US. Those are some of our upcoming events. We've been working very hard this summer to put together an exciting program of panel discussions, tech talks, and hopefully you can attend some of them. Those are just a few of them that we'll be hosting in the next weeks. So make sure to stay tuned to look at what we post on Facebook, Twitter, Meetup Group, and so you can secure a seat before it's full. We'd like to thank our sponsors tonight, but also NetApp for hosting us, as well as our partners. A few words about The Hive. Usually our founder, TM Ravi, is attending this event. He's here every week. Every time we have an event. He couldn't come tonight, but I'll say a few words about The Hive. The Hive is a data-driven startup incubator based in Palo Alto. So we incubate startup developing applications for businesses. And we invest from 1.5 to 3 million in each one of these startups. And we are kind of a low volume, high touch incubator, which means that we invest a lot of funds, but we also work very closely with our startups. We have an in-house data scientists, in-house architects, marketers that are really here to help our startups grow. And those are some of our startups. Of course, we are hiring. And remember, tonight, use our hashtag HiveData to continue the conversation online. And I'm going to introduce very shortly, sorry I have my notes, our friend Skip, Vernon. His name is Vernon. He's a doctor of minor. He has over 40 years of R&D experience in cellular biology, immunology, and biotechnology innovation, and has made broad contributions ranging from basic immunology to technology and product development in the areas of immunology, cell biology, and flow cytometry. Sorry for my pronunciation. Thank you so much for reaching out to Mike Snyder, and it's a great pleasure to have you co-host the event tonight. I know you'll introduce Professor Mike Snyder, so I'll give you the microphone. Thank you.
Thank you, Pashu. I'm going to keep this very short because I'm eager to hear the talk from Mike. We've all heard a lot of buzz about personalized medicine and how it's going to move us away from reactive healthcare to more strategic and proactive care focused on wellness. So we're fortunate tonight to have one of the experts in this area, Dr. Mike Snyder. He's had a chance to talk to him briefly before I came here, so I learned a lot more about what he's doing. But he's chairman of Department of Genetics at Stanford, and he's also director of the Genome Project there as well at Stanford, and he's a principal participant in ENCODE, which is an interesting worldwide consortium of scientists dedicated to providing information about the human genome and its function. So we're very fortunate tonight to have him talk more about his area and give us some real insights into the concepts of personalized medicine and where it can go. So Mike, please.
I think I'm wired here.
Well, you're already wired.
Can you hear me all right? Yep, great. Well, it's really a great pleasure to be here. I'm actually even thinking about my next startup already. So maybe it'll be quite timely. We'll see how it goes. Sorry, no, somewhere I put a... sorry about that. Oh, hello? Well, there we go. All right, thanks. So I am going to tell you about some of my thoughts in this area. There's a lot of different subjects. I know you might be more IT, big data types, and we can use this as a sounding board to let the discussion go wherever it may go. So I'm going to start with a premise that I always like to start with because I really think it is the foundation of everything we do. And it really, if you think about it, it's the substance of where big data is going to hit in the whole healthcare profession. And that is our health, or I like to refer to it as our health state, because it's a dynamic state where we toggle between healthy and disease states as we go through life. It's really a product of our DNA, our genome, if you will, which is our collection of DNA, and all the various things we're exposed to. So the food we eat, that beer you're drinking right now, the life stresses, food, exercise, so your physiology, all this, if you will, it's input along with your DNA that leads to a certain output, which is a health state. And again, it's a dynamic state, and I think we all know this intuitively, this is what happens, but I would argue a goal of us, and I think this means all of us, is to understand this equation so well that we're predictive. That really is the goal of medicine, to be able to know with your DNA sequence what various things you're exposed to, what the possible health risks are. And this really is a probabilistic determination. It's not going to be absolutely deterministic. That is, if you eat, that certain meatball, you will guarantee get, a heart attack. It doesn't work exactly that way, but it will increase your chances at some frequency. And so again, that's what we'd like to understand in probabilistic terms. And this idea isn't new. It's been around for a long, long time, and in fact, it was quite popularized, as you may know, by this movie here, Gattaca. How many of you have seen this movie? Okay. How many have not seen this movie? Oh, quite a few. You can literally buy it for $5 at Walmart. And so I bought a whole bunch and gave them out. And it's very interesting because I saw it not so long ago. And a lot of the concepts that are in this movie, well, I don't want to be a spoiler, but anyway, a lot of the concepts which relate to DNA and the occupation of Ethan Hawke is, those concepts are still very relevant today, and we've talked about it quite a bit. So anyway, that's the idea. With your DNA, you can predict with certain, in this case, occupational exposures, what your outcome might be. Anyway, this first part of the equation, understanding your DNA is now very solvable, you may know, because of the revolution of DNA sequencing. And that started in about 2004, 2005, and then it really took off in the mid-2000s. And it's plummeted about 10-fold every 15 months or so. And it's going down a little bit more slowly in the last year, partly because there isn't as much competition as we'd like to see. But in general, it's still heading down. And it's at the point where we now pay $1,400 for a human genome sequence. Okay? So that's less than a cheap used car. So you can buy your sequence. Now that's just the sequence. The interpretation, the way we do it for a healthy person, it's about $15,000. And we'll see why in a minute. So there's still, and this is an area of opportunity from the business side, and I'm biased because I found a company that does do this sort of thing. But basically that is a bottleneck. Interpreting genomes for healthy people is not trivial, trying to predict disease risks. Now, just to get everyone oriented, I just want to say a few words about DNA. So your DNA is comprised of 4 letters, if you will, and they're arranged in a very particular sequence. We have 6 billion of these letters. So when you get your genome sequence, the mission is really to get that order correct for all 6 billion letters, and in our case, the genomes are packaged in the chromosomes, half from our mom and half from our dad. So when you sequence the genome, you want to get this order correct. And for the most part, the order of the sequence is pretty similar amongst all of us in this room. But there are differences, and in fact, there are these what are called single nucleotide changes, or single nucleotide variants. And there's about 1 in every 1,200 between you and the person sitting next to you. Okay, so there are differences. That's the frequency, 1 in every 1,200. And then there are other more serious events, short insertions and deletions. Some of these can be quite serious because they kill genes, if you will. We have about 20,000 genes embedded in our DNA. Some of these short insertions and deletions, so there'll be segments that are deleted or grow a little bit, and there'll be about 500,000 or so of those. The exact number hasn't been pinned down to many people. And then there are actually, believe it or not, large structural variants. They're called large chunks of DNA that will differ between people. They can be up to 1 million bases long. Typically, many of them are, guarantee you'll have a few of them in you that's different between you and the person next to you by hundreds of kilobases. So there'll be big chunks of DNA that differ. So we're all quite different from one another, and we're different by large numbers overall. Okay? And so the goal is when you get your genome sequence, we actually don't sequence, the technology is somewhat limited. We don't sequence these de novo, that is, put together your sequence in a string. Typically it's done by making short reads, they're called, and then matching up to a reference, and you try and find these variants. So when you get your genome sequence, at the end of the day, you're getting these variants determined relative to a reference. And this is typically how it's done. Okay? So I won't get into technology any further than that, other than to say that that is the name of the game, but that's an area of opportunity as well. Now, because it's gotten pretty cheap to sequence genomes, it used to be $1 billion to sequence the very first one. Lots of famous people have gotten their genome sequence. So these are some of the first two: Jim Watson, Craig Venter, and Ozzy Osbourne, and Glenn Close, and other famous people have gotten their genome sequence. And just out of curiosity, has anyone here gotten their genome sequence besides me? Yeah. Well, only one in the back, too. So I think there'll be more to come in the future. Okay? So you can get your genome sequence. Now, it's what you do with it that's probably important. Nonetheless, from this, the fact that genome sequencing is here, the fact that it's becoming cheaper, means it's having a huge impact, as you might imagine, in the medical arena. And I listed some of the areas up here. We're going to get into this in a little more detail in a minute. Understanding and treating disease, so cancer is a genetic disease, we'll get into that. Understanding undiagnosed and mystery diseases, prenatal diagnostics, these are all areas where genome sequencing is having huge impact already. Other areas that are starting to have some impact is in pharmacogenomics, so predicting your drug sensitivity. I won't get into this much today, but if you don't learn anything else from your genome sequence, you'll learn about your sensitivity to certain drugs like warfarin and other sorts of drugs. I guarantee, at least we've done a fair number of genomes, I'd say on average for every person we've found at least 6 useful pieces of information. I hope you'll never need those 6 pieces of information, because you'll never need warfarin, but if you do, at least you might know a starting concentration or what possible side effects are. Like, I take statins, I have high cholesterol, I'm a great responder, but I know what statins I should take for my genome sequence. And that relates to this last topic, which is this is generally geared towards sick people. What if you're a healthy person? Can that be valuable? Or can getting your genome and other omics information be valuable for managing your health? And that's where I actually want to spend a fair amount of time. Let's just jump in on some of these first topics so you can see where this is having impact and some of the opportunities here. And there's room for, I think, many startups, more startups in this area. So cancer genome sequencing is arguably the area where there's the most impact from human genome sequence. And the reason for that is cancer is a genetic disease, both in terms of predisposition for cancer, it can run in your family, and also when cancer is acquired, almost invariably genetic mutations have happened to help drive the cancer. So cancer is cells growing uncontrollably, and mutations appear that basically drive the cells to do that. And it's arguable, but typically it's thought there's as many as 10 or 20 different driver mutations, mutated genes, that will cause cancer to grow uncontrollably. Now, what's been discovered by now sequencing thousands of cancers is that every single cancer is unique. So if you've seen one cancer, you've seen one cancer. And it won't necessarily be the same as the next ones. Now there are common pathways that are hit, and this lists a few of them over here, and there are preferences for things. So melanoma, 60% of the individuals will have a mutation in a gene called RAF, very specific mutation that causes them to grow. It's out of control. But in other cases, it's not so predictable. And to give you an example, we do sequencing of cancer genomes, and typically the way it's done is you'll sequence the cancer tissue along with normal tissue, and you look for those somatic changes. Those are, sorry, I forgot to say, somatic changes are changes that occur. You weren't born with them, you acquired them through life. And so in the Cancer Genome Project, you'll sequence the cancer tissue along with the normal material. And you look for those somatic changes and try and guess which ones will be driver mutations. Typically, a typical cancer will literally have thousands of mutations in it, and so it's actually a challenge to do this. But nonetheless, I can tell you we've done quite a few cancer genomes now so far. This is one example where this is an individual with metastatic colon cancer. With Hong-Li, we sequenced his genome, and this individual is amplified, meaning he had increased copies of two regions of the genome, EGF receptor, and this one here, it's a protein kinase. Now this gene is well known to be mutated, especially at that time, in non-small cell lung carcinoma, but not so much in colon cancer, very rarely actually, 2% of the time. But this individual was mutated in this cancer. Why was this information important? Well, there are two drugs that are known to hit this, the product of that gene, the EGF receptor. Two drugs on the market, not approved for colon cancer, they were approved actually for non-small cell lung carcinoma. But you can actually use that drug in this individual, and so this individual is on one of those two approved drugs. So this is how the cancer arena is unfolding, which is people are sequencing genomes because everybody's different. You try and guess which ones are the driver mutations, and then pick drugs that will actually hopefully help that individual. Now, just to give you a personal example of this, so my mother-in-law actually had ovarian cancer fairly recently. It had a long time ago, it came back. She was at Sloan. I said I wanted to sequence her genome. There were some complications getting her, thanks to the transferring material. Welcome to the medical profession. So they didn't exactly give us the material, at least not right away, but they did go in and do genetic tests, which I'm not so sure they were going to do had I not said we wanted to sequence her genome. So they went in and they looked at a lot of obvious loci. There are companies that will do more targeted stuff, and so they did some targeted tests, and it turns out that, in fact, she was found to have a mutation in, it's called a MEK kinase, and actually there was a phase 3 drug for that, and in fact she took that drug and responded well. So she actually, her health was in pretty serious decline, and then with this drug she actually got a lot better, although like virtually all cancer patients, resistance did come back. Okay, why did resistance come back? Well, cancer is fairly heterogeneous, right? When you have a tumor, you have lots of different cells in there, millions of cells that are cancer. Some of them are resistant. And sometimes the mutations aren't always the same in the different cell types. It depends when that mutation arose. So really the way the field is moving towards is actually to get multiple drugs now to any individual cancer. And so it'll be like AIDS, where you use combinatorial therapies to try actually target individuals now, not just with one drug, but with multiple drugs. And so, again, there are some cancers like CML, where with certain mutations, those individuals will show resistance for 5 years, will show good health for 5 years over others where mutations come back, or resistance comes back very quickly, and people pass quickly. But we don't know all the rules yet, so there's a huge opportunity here for information science to have impact by trying to understand the rule. There are millions of people who get cancer in the US every year. They're all getting treated, and when nobody collects that data in any uniform fashion. So it's just willy-nilly out there in the hospitals. Everybody's saying, well, we should do something about it, and nobody's really doing anything about it. They always raise issues, well, it's privacy, blah, blah, blah. But it's really a matter of just not getting ourselves organized. And I think that's what, there is a big push to try and do that. So imagine having millions of patients with millions of medical information, with millions of drugs, or hundreds of drugs, all put these individuals. We could do this a lot more systematic and informed than we do now, because the way it's done now is someone gets cancer, there's always, here's the first drug you should use, because that's what's given to ovarian cancer patients, even if it's not the best treatment for that cancer. So we give medicine right now to people based on tissue type, which we now know is the worst way to classify cancer. It's not tissue is the best way, it's mutated genes is a much better way, or gene expression signature. So we really need to be pushing this in the proper direction. Okay. Other areas where genomics is having a huge impact is in solving mystery diseases. So there are kids born every year with undiagnosed diseases, or problematic diseases, I should say. And half the time they can figure them out, and half the time they can't. And 4% of kids that are born each year will have an undiagnosed disease that's not figure outable easily. And so what parents do is they turn to genome sequencing to try and solve what's wrong with their kid. Okay, by the way, that number is often said 8% by the time people reach adulthood. People have some sort of genetic defect that this information can be useful for. Anyway, so nonetheless, it's 4%. It doesn't sound like much, but there are something like 10 million babies born. I can't remember the number. Someone here probably knows.
4 million
12 million
per year.
per year? 4
4 million.
million per year. Thank you. So 4 million babies per year. 4%, that's still a large number. Okay. And believe it or not, some people say from a business side that actually this market is larger than the cancer market from numbers. The numbers that I heard were this market is estimated to be about $13 billion per year, whereas cancer is thought to be on the order, from the genome sequencing side, something like 8.4 billion are the numbers I had. All right. Anyway, let me go through an example. Here's a case where there was actually, this is someone who was a senior in the life technologies company, and they had 2 kids who were twins who actually had problems at birth. And just like you tell an interesting lesson, they were born as so-called floppy babies, and they had a number of abnormalities. And there were, the mom actually figured this out by reading the literature. She figured out that she thought the kids had dystonia, and they often treat dystonia kids with this drug called L-dopamine, which is a neurotransmitter. And they showed a lot of improvement, but they weren't completely cured. They were just in much, much better shape, and they did pretty well. This is the very twins, and this is not our work, this is from Richard Gibbs and others. And so what they did was they sequenced the children and the parents of these, and they found a mutation actually in this pathway that makes dopamine. And this is why they felt they were on the right track, because it was in the pathway they were looking for. But it turned out this pathway not only makes dopamine, it makes serotonin. And these kids, they were actually, their health was starting to decline. They were teenagers. And so that's when they sequenced their genomes, discovered, in fact, that they're defective in this pathway, and you don't give serotonin, but you can give a serotonin precursor to the kids. And so, in fact, the kids were given the serotonin precursor, and by all visible means, they actually seem to be cured, or at least managed in their health. So the point is that in this case, it did gene sequencing the kids, gave them information that was actionable. Now, I can tell you we've done quite a few of these, and most of the time we don't find the cause of the mutation. And not only that, I don't think we've ever found a case that's been actionable therapeutically, although I would say it's still been actionable in other ways. And this is a very typical example for one we solved, which is there was a child born with, it's a more non-specific effect, that they had developmental delay, intellectual disability, hearing loss, problems all over the place, and it was much more difficult to ascertain exactly what was wrong with this child. So we sequenced the father, mother, and the child, and we're looking for mutations where both genes are inactivated in the child and only one in the mother. That's the simplest model. And this is a challenge, yeah, there are millions of variants. Many of them are private that you think would be more specific for the child. And I did say, but there's about 10,000 sitting inside the genes themselves. So it's a lot of mutations. Which one's causing the problem? That's the challenge you face. So we sequenced this, and for a variety of reasons, we narrowed it down to these 7 genes that we thought were possible candidates for causing this child's problem. And this is a very typical example. Occasionally we can say, oh, that's the likely gene, because it matches the defect that we're saying. In this case, it could have been any one of 7. A lot of people like that one. There's some reasons why that was less attractive. At the end of the day, the way this one got solved was 8 months after we sequenced this genome. There was a second case of a child with somewhat similar symptoms, and they sequenced their genome at Duke. And of all things, they posted on a blog that their child had a mutation in this gene, NY1. And they posted on a blog, and then that match got made. So it's crazy, right? The way this disease got solved was somebody searching the blog with these variants and found this match. Okay, and said, well, they're pretty similar. Maybe they're the same, so they compared notes and looked at it more. It's very clear that is the cause of the mutation. There are now 16 kids running around who have very similar symptoms, who actually all have mutations in NY1. So, in fact, that's how this got solved. So the lesson out of this is A, it's hard. B, this is a crazy way to be sharing information to figure this stuff out. So there's a lot of interest in setting up databases now so you can share this. And again, there's a lot of issues associated with privacy. I can guarantee the parents, by the way, could care less about the privacy. They always want to solve the kid. The doctors are the ones who care about the privacy and others, but as far as I can tell, the parents never seem to care. Okay. So anyway, so there's a lot of efforts going on in this space. So overall, our success rate for identifying cause of mutation is about 25%. Depends on the type of child we're looking at. And so we do need these large databases where you can share this information so that you can make these NY1 matches without having to search blogs and have the information in a very useful fashion. Because a lot of information that's in the public databases is garbage too. So you don't want to be making wrong diagnoses. Like we would say, for one of the most highly respected databases of gene variants, 8% of the time they've entered the wrong sequence in the database. So we know that one's off quite a bit. So anyway, we need to have more of this, and we need to set up downstream assays, and that's something our lab is doing. So for the individual I just mentioned, this person actually, we made stem cells from them. We're actually trying to cure the mutation, if you will, in culture. Because they have a stop codon and frame shift. This is a little jargony, but they're actually drugs that can be used to treat that. We could see whether they'll have any effect starting with the kids. I can tell you, even in this case, that even though I'm not sure that we'll ever help that child with this information, it does help people in other parents in other cases. That is to say, parents who are thinking about second kids actually do use this information. They almost invariably say, I love my child, but I sure would like to have one that doesn't have the same problem. And so they'll actually turn to doing in vitro fertilization and testing that way to see whether to avoid having a second child like this. So the information may not help this individual, but could help future generations. Yeah?
When you say we've made stem cells, who's
Right.
the we there?
Well, we as our lab.
Research group? Okay.
Yeah.
Not a company that you've associated with, your research group.
Our research group, yeah. I don't know if there's a personal stem cell company. I do think in the future we will all have stem cells ourselves that we will test drugs on. Because why say, yes, I think I'm sensitive to that drug, but you might as well test it because you can make cells of yourself. And I can tell you we have 8 lines in the we're doing all kinds of fun tests with those lines. Yeah.
So far you're talking about single variations. Is that always what you expect to find is being positive?
Yeah, that's a great question. So you said we're only looking at single variations, and we're generally following the model that these are recessive mutations. And this may be why we're not always solving these, where each parent has one gene that's been mutated, if you will, and then the child gets both mutated copies. And that's typically how we find it. There's a few cases where you can look for what's called a dominant mutation. So if the parents are healthy and the child comes up with a problem, and it's not this recessive mutation, it could be a single mutation that causes the problem, and then the child probably got it spontaneously as opposed to from one of the parents. The parent should show it if they had that, but not always. It's tricky business. It's not an easy situation. This is why we need to share this information better. Okay. And then there can be, by the way, some of these can be due to big deletions, and that's been shown, especially a lot of the intellectual disability disorders often have large deletions or insertions associated with them. And for complex diseases like autism and things, the individual mutations in general aren't thought to be strongly causative. It might be a combination of mutations, and that's maybe what we're getting at. That's an area we work at very, very hard. We have a very interesting story around autism in that respect. All right, so anyway, these are some of the lessons from these strong cases. I've probably gotten behind, so I'll simply say the other area where genomics is having huge impact, you may know, is in fetal testing. So it turns out that when mothers are pregnant, they have a lot of DNA from the fetus circulating in their blood. It starts out as about 4%. By the time of birth, it's about 13%. And you can actually take that blood from the mother and sequence it and figure out which one's the kids. And this is now an FDA-approved test for Down syndrome. It's very easy to find these large aneuploidies, these large chromosome rearrangements. So it's now approved test for Downs, chromosome 18 and chromosome 13, extra copies of those, which do cause severe problems you can screen for. And you can actually do much, much better than that. You can actually take over, but you can see smaller effects as well. And quite frankly, in the future, you'll be able to sequence effects already shown, sequence the entire child's DNA from the mother's blood. Okay, so that's going to set up all kinds of interesting issues as well. Okay, but no one will have a problem with doing these sorts of tests now. It's much, much safer than amniocentesis and other methods. And this is a case of just detecting not a whole chromosome, but part of a chromosome that there were extra copies in it. So this is an area that interests us. This is this idea, well, what if you're healthy? Can getting your genome sequencing be valuable to you? And this is something you should all think about because you all will have the opportunity of getting your genome sequenced. And the idea is the following, that you get your genome sequenced and then be able to use this information to predict your disease risk, minimally diagnose disease early, monitor disease states, possibly treat with the pharmacogenomics, and minimally, like undiagnosed diseases, understand disease states. Now, I'm a big believer we should go well beyond genome. We should also bring in other kinds of omics as well, because right now, if you go to a doctor, they measure about 15 things in you. And there's no reason our lab, which does proteomics and lots of other omics shown here, and in principle, you can measure tens of thousands of things. And so what we did when I moved to Stanford, which is now 5 years ago, we set up assays to be able to measure all of this in a single person. So in addition to getting your genome done, you'll get what's called your epigenome. This is your DNA methylation pattern on your genome. All your RNA molecules, this is what your genes make RNA. They also make proteins, so there's a collection of proteins called the proteome. So this is for the aficionados, but we measure this using mass spectrometry out of the blood. We also follow the these immune stimulators called cytokines. We follow 63 of these with a hydrochloric acid. We've spent a lot of time on this. It's a very underdeveloped area. These are your metabolites, your collection of metabolites. We follow about 25,000 metabolites total in urine and plasma. We also follow the antibodies you make against viruses and yourself in here. And lastly, most recently, we've added on something called the microbiome. Many of you may know there are more cells in and on you that aren't you than are you. Meaning to say you have 10 trillion cells, but you have 100 trillion bacteria inside you digesting your DNA, digesting your food, I should say, making vitamins, things that are absolutely essential for you. And they're all over your skin, they're everywhere. So a lot of it's good and occasionally it's not so good. So anyway, we measure 5 different microbiomes: gut, urine, nasal, tongue, and skin, and how we're trying to add vaginal. So initially when we set this up, we were following 40,000 molecules in the blood. Now we literally make billions of measurements when you count all the microbiome and the DNA methylome and all this stuff. And I'm gonna, because I've got later, I'm gonna zip through some of this, but as a proof of principle, we actually tested this out on me of all things, so you may have heard this story. And it was really, it was just a pilot study. I didn't think I would be very interesting. But the day we started the study was here, and it turns out I had a rhinovirus infection, common cold. So we took blood that day, 4 days later, 21 days later. And then what you'll notice is we've been doing this first of all for 4.5 years. And I did my 81st time point last week. So we've been sampling for quite some time. And I've been through 6 viral infections. You can tell I have little kids. So I've gotten sick twice a year until recently. They're getting older. And what we do is you can see we do dense sampling whenever I get sick. There's a respiratory syncytial virus, very common in kids, not so common in adults. When adults get this, they get quite sick, and that was true for me. I was out for 3 days. We took blood that day, next day, 2 days, 2 days, 3 days. So we do dense profiling when I'm sick, and every 2 or 3 months or so I'm healthy. And so I've been through all these different infections, except for flu. And I rarely get a flu vaccine, my family does, so I think I'm getting herd immunity, if you will. They're protected, and I get the piggyback protected. This one I'm going to come back to later. It's very interesting. It's a double infection, it's coronavirus infection. So anyway, we've been profiling me during this time. And I'm going to jump through this, just to get to the main conclusions. But we started by sequencing my DNA, which made lots of predictions and helped tell us how do we interpret a healthy person's genome. We made some predictions that aren't accurate based on the literature that's out there, like I'm supposed to have aplastic anemia, which I don't. And it turns out we don't think the literature is quite right on that one. But there are other areas that were quite interesting. So we can predict complex diseases using complex algorithms, good area for business, by the way. And you can do this literally is what 23andMe does. You have markers of disease and you add them up. And you do this for hundreds of diseases. I'm just showing you the top of my list. These are the things that I'm going to either increase or decrease this for. So as a European male, this triangle is my starting point for getting one of these. Again, it goes on for hundreds of diseases. And this yellowish-orange line means increased chance of getting one of these. Blue means decreased chance. And my profile actually matched my family history incredibly well. That is to say, nobody in my family has ever been overweight. I'm a decreased risk there. Everyone on my father's side, except for one, has died of heart failure, including my father's side. Increased risk there. I told you I was on statins because I have high cholesterol that shows up here and here. So most of this makes sense. There were two surprises. One is here, increased risk of basal cell carcinoma, which I was not aware from my family history, but we had a family reunion about 2 years ago. I have 2 brothers, 3 sisters. They all came out but one sister. So we took blood from everybody so we could profile them just like me, of course. And I just happened to ask 2 brothers and a sister, what do you know about basal cell carcinoma? So I'm at risk for this. And it turns out each of them had lesions, which I wasn't aware of. And the reason I wasn't aware, you don't talk about basal cell carcinomas around the dinner table, right? It just doesn't come up. So I'm a big proponent that your genome is a better record of your family history than your family is, because it's more truthful in what it's telling you.